Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine the incredibly delicate dance of creating life,
Right.
now guided by, well, a digital hand from thousands of miles away.
It's pretty amazing to think about.
Today, we're plunging into the fascinating story of the first baby born through a completely automated, digitally controlled method of injecting a single sperm into an egg.
The procedure known as intracytoplasmic sperm injection.
Exactly. And doing it, like, automatically.
That's right. Think about this. Artificial intelligence and robotic precision are now intimately involved in, helping people build their families.
Yeah.
You shared some really compelling information about this development, and our goal today is really to extract the most important and maybe surprising insights for everyone listening.
Definitely. We want to really understand what this automated intracytoplasmic sperm injection system means, for the future of fertility treatments.
Right. One of the possibilities.
What exciting possibilities does it open up, and what are some of the hurdles that still need to be overcome?
And to get to the heart of this, we'll be looking closely at the detailed research. It was published in the journal Reproductive Biomedicine Online.
Yeah.
And we're also pulling from a related article from a health news outlet that highlighted the significant achievement.
Okay, great. So let's start with the basics of the research then. What was the main question the scientists were trying to answer here?
Well, the fundamental question was whether a fully automated system, one that's controlled digitally and can even be operated remotely, could successfully perform all those really intricate steps of intracytoplasmic sperm injection.
And actually result in a baby.
And ultimately lead to a live birth.
Yeah.
That was the big goal.
So it sounds like a real leap forward.
Yeah.
So who is behind this technology? And, how do they actually build this automated system?
Right. Well, this was a collaborative effort. It involved experts from a company called Conceivable Life Sciences.
Okay.
And they had teams in New York and also Guadalajara, Mexico.
An international team.
Exactly. The key individuals who really spearheaded this work included Dr. Jacques Cohen, who's a very experienced embryologist,
Right.
Dr. Alejandro Chavez Badiola, the chief medical officer, and Professor Gerardo Mendizabal Ruiz, who's a leading engineer.
So a real mix of skills.
Oh, yeah, definitely. It was truly a blend of biological and technological expertise.
So what exactly does this system look like? What can it do?
The system they developed is essentially this sophisticated workstation. Think of it like that.
Okay.
And it's designed to automate all 23 individual steps involved in the standard, the manual intracytoplasmic sperm injection procedure.
23 steps. Wow.
Yeah, quite a few. And what's truly novel is that each of these steps can be controlled in two ways.
How so?
Either independently by artificial intelligence or remotely by a human operator, just using a digital interface.
That's a big shift from how it's typically done.
Oh, absolutely.
Because currently, isn't intracytoplasmic sperm injection a very hands-on procedure, performed by highly skilled embryologists?
Precisely. Every single intracytoplasmic sperm injection procedure performed today relies on the manual skills and, frankly, the expertise of embryologists using these microinjection systems.
Right.
And Dr. Cohen himself, one of the researchers, has noted that, the performance of embryologists can naturally vary from one person to another.
Sure, that makes sense.
So he believes that automating this entire process offers a potentially transformative solution.
How?
Well, by leading to greater accuracy, improved efficiency, and more consistent results. Basically, by reducing this inherent human variability.
And maybe taking some pressure off the embryologist, too.
And even alleviating some of the workload. Yeah, absolutely.
That makes a lot of sense. So let's delve into the nuts and bolts of it then. How does this automated system actually work? What are the key pieces of tech?
Okay, yeah, there are several really fascinating technologies at play here. First, they use artificial intelligence to select the sperm.
AI for sperm selection.
Yes, it's a system they've named SID. The artificial intelligence is programmed to identify what it determines to be the most viable sperm cell,
Okay.
and then it uses a laser to precisely hold its midsection.
Wow.
Professor Mendizabal Ruiz even suggests that the selection and immobilization could potentially be more accurate than what a human eye and hand can achieve.
So the laser isn't just for holding the sperm still. Is there more to it?
That's right. Laser technology has a further role. After the initial holding, the system uses 1 or 2 additional laser pulses,
Okay.
very precisely targeted at the mid-tail region of the sperm, just to ensure it's completely still for the injection.
Got it. Immobilized.
Exactly. Then they employ computer vision.
Computer vision, like teaching the computer to see.
Sort of, yeah. Think of this as the system's ability to see and interpret what it's looking at.
Yep.
It visually identifies and accurately pinpoints the different components. The sperm's head and tail, the outer layer of the egg that's called the zona pellucida, the egg's membrane, known as the ulema, and crucially, the exact position of the injection needle as it enters the egg's cytoplasm.
It's like the system has its own eyes and, well, the intelligence to understand what it's seeing.
That's a good way to put it.
So what happens when it's time to actually get the sperm inside the egg? How does it manage that?
For that critical step, they use something called piezo-actuated pulses.
Piezo-actuated. What's that?
They're incredibly tiny, precisely controlled mechanical taps, basically.
Wow, little tap.
Yeah, and they help the injection needle gently penetrate the ulema, that egg membrane,
Right.
making it easier to deliver the sperm inside without causing damage.
Very delicate.
Extremely. And the entire system, it has motorized components, things like the microscope stage, the objective turret.
Okay.
All of these are under digital control, which was what enables the remote operation, you see.
So that's how they do it from afar.
Precisely. These motorized parts allow for the fine, automated movements necessary for the procedure to be controlled from, well, potentially thousands of miles away.
This is incredibly detailed, amazing stuff. So how was this system actually used with a patient? Was this just theoretical?
No, this was a real clinical application. The treatment took place at Hope In Vitro Fertilization, Mexico, down in Guadalajara.
Okay.
And it was conducted under the careful ethical review of a board as a pilot investigation.
Right, a first step.
Exactly. The patient was a 40-year-old woman who had previously experienced an unsuccessful in vitro fertilization attempt.
Oh,
And for this particular treatment cycle, she received eggs from a donor.
got it. And how did the automated system perform compared to the standard manual procedure in the specific case? Was there a comparison?
Yes, there was. The donated eggs were divided into 2 groups.
Okay.
5 eggs were assigned to be fertilized using the automated intracytoplasmic sperm injection system. And this was all done remotely.
Remotely, meaning?
Meaning the operators controlling the system were located not just in the same clinic, but also in New York.
New York. But the clinic was in Mexico.
That's right, about 3,700 kilometers away.
Wow. That really emphasizes the remote aspect. And the other eggs.
The other 3 eggs served as a control group. They were fertilized using the standard manual intracytoplasmic sperm injection procedure, the usual way.
Okay, a direct comparison. How did the time taken for the automated procedure compare to the manual one? That seems important.
It is. And on average, the entire automated procedure took about 9 minutes and 56 seconds per egg.
Nearly 10 minutes. And the manual?
That was considerably longer than the manual control group, where it took just 1 minute and 22 seconds per egg.
Oh, quite a difference.
A big difference, yes. However, the researchers have explained that because this was an initial experimental setup.
Right, first thing out.
They fully anticipate that the time taken by the automated system will decrease significantly as the technologies further developed and, optimized.
That makes sense. Speed and efficiency are obviously so important in these procedures. So, okay, what were the key results in terms of fertilization and the development of early embryos?
Well, the fertilization rate in the automated group, the remote group, was 80%.
80%. 4 out of 5 eggs.
Exactly. 4 out of the 5 eggs were successfully fertilized, which was actually quite comparable to the manual control group.
What was their rate?
They achieved a 100% fertilization rate. All 3 eggs were fertilized.
So pretty close results there.
Very close. And importantly, in both groups, 2 usable blastocysts were created.
Blastocysts being those early stage embryos, right?
That's right, the ones with the potential to develop into a pregnancy. 2 in each group.
So the initial stages showed promising results in both approaches then. What happened next with these embryos? Were they transferred?
Yes. A fresh embryo transfer was performed using one of the blastocysts from the automated group.
Okay.
But unfortunately, this did not result in a pregnancy.
Ah, okay. That can happen, of course.
It can. However, they also had another blastocyst from that automated group that was cryopreserved.
Frozen, you mean?
Yes, frozen and stored. And when this frozen blastocyst was later thawed and transferred, along with a procedure called assisted hatching, which helps with implantation,
Right.
it resulted in a healthy live male baby being born at 38 weeks of gestation.
That's remarkable. The very first baby born using this fully automated, remotely controlled system for intracytoplasmic sperm injection.
The very first, yes.
Wow. What does this success really tell us about the future of fertility treatments?
Well, the implications are frankly quite profound. Firstly, automation has the potential to significantly increase the precision and consistency of the procedure.
How so?
By reducing the direct human involvement, the aim is to minimize those natural variations that can occur in manual procedures.
Right, the human factor.
Exactly, which could ultimately lead to more consistent and predictable outcomes for patients.
So it's about bringing a greater level of standardization to a very delicate and, let's face it, complex process.
That's a key part of it, yes.
What about the efficiency? You mentioned it took longer initially, but is there potential for the automated system to become faster?
Absolutely. While it was slower in this, initial experimental phase, the expectation is that as the system is refined and becomes more autonomous, it will be able to perform the procedure much more quickly.
Faster than humans even.
Potentially even surpassing the speed of manual intracytoplasmic sperm injection in the future. That's the hope.
That could really change the landscape, couldn't it? Just in terms of the number of treatment cycles that could be performed.
It really could. And there are other potential advantages, too.
Like what?
Well, Dr. Cohen suggested the possibility of improved egg survival and even optimizing the precise timing of the sperm injection.
Timing is critical.
Very critical. And that could have positive effects on the outcome. Furthermore, just by automating these physically and, let's be honest, mentally demanding steps.
Yeah, it must be stressful.
It could also reduce the workload and stress experienced by embryologists.
That's a huge point. And the ability to operate remotely. That seems like it could really expand access to specialized skills, wouldn't it?
Exactly. Think about it. Remote operation means that highly skilled embryologists could potentially oversee procedures in multiple locations simultaneously.
Across continents, even, like in this study.
Even across significant distances, yes. This could make expert care available in areas where there might be a shortage of experienced embryologists.
Right.
And the researchers also propose that individuals operating the system digitally might not need the same extensive prior experience in micromanipulation as those performing the manual procedure, so it could broaden who can provide this specialized care.
It sounds like it could really make this type of fertility treatment more accessible potentially to more people.
That's certainly one of the major potential benefits.
What about ensuring consistency in how the procedure is performed across different clinics? Does automation help there?
That's another key advantage they highlight. Automation could lead to a greater standardization of the process across different clinics, different regions.
So same quality of care, hopefully everywhere.
That's the idea. It could help to ensure a more uniform quality of care for patients, regardless of where they receive treatment.
Right.
And looking at the bigger picture, this development is a significant move towards, well, the full automation of the entire in vitro fertilization laboratory.
It's not just this one step.
No, other processes like embryo culture, cryostorage, sperm assessment, they're also seeing increasing levels of automation. This fits into that larger trend.
It paints a fascinating picture of where fertility treatment might be headed. But, with any new technology, there are always challenges, limitations.
Of course.
What are some of the limitations that were noted in this study? Can't all be perfect yet.
No, and you're right. It's crucial to have a balanced view. One of the main limitations at this point is, as we discussed, the time factor.
It takes longer right now.
The automated procedure currently takes longer than manual intracytoplasmic sperm injection. That's partly due to the experimental nature, the need for careful monitoring, data collection.
Makes sense. And was the system completely independent in this case? Or did humans have to step in?
Not entirely independent, no. The study reported that on-site human intervention was necessary for the initial setup of the system. And also on one occasion to troubleshoot an automated step that didn't quite work as planned.
So not fully autonomous yet.
Exactly. So while it's a significant step towards full automation, it hasn't yet reached a stage where absolutely no human involvement is required on site.
The remote operation also brings up some, like, practical considerations, doesn't it? Like the internet connection.
Absolutely, that's a big one. The system's functionality relies heavily on a stable and high-speed internet connection.
Needs good broadband.
Yes, the researchers specified the need for broadband with minimum upload and download speeds. Issues like latency, delays in communication could potentially pose risks during the remote procedure, just given the incredible precision required.
That's a really important point. Even a tiny delay could be critical in such a delicate procedure.
It could.
What about the natural variations? Eggs and sperm aren't all identical. How does the robot handle that?
That's another key question and maybe a limitation for now. It remains to be seen how well the automated system can adapt to the natural diversity and variability that you see in eggs and sperm from different individuals.
Right.
Manual intracytoplasmic sperm injection often requires the embryologist to make these real-time adjustments based on those unique characteristics.
Judgment calls.
Exactly. It's still a bit unclear if the automated system can optimally handle all of these diverse scenarios just yet.
And in this initial case, this first baby, was the team involved smaller than what's typically needed for the manual procedure, or bigger?
Actually bigger. The researchers noted that the number of human operators involved in this initial case was higher than what would typically be required for a manual procedure.
Oh, why was that?
Well, it was because engineers were working really closely with the embryologists during this experimental phase. Lots of oversight and collaboration needed.
Ah, okay. That makes sense for a pilot study. And I suppose the results from just one successful birth, that needs to be confirmed with more research, right?
Oh, absolutely. And the researchers themselves emphasize this very strongly.
Yeah.
They state clearly that the system's validity and its potential for broader use need to be confirmed through larger studies involving a much greater number of treatment cycles.
So while this is a huge milestone, there's still more research and development needed before this becomes like standard practice.
Definitely. It's a proof of concept, a very exciting one, but more work is needed.
So what did the experts involved in the study have to say about these findings overall? What was their takeaway?
Well, Dr. Cohen, for instance, emphasized that this automation, in his words, represents a transformative solution that promises to enhance precision, improve efficiency, and ensure consistent outcomes. And he linked that back to reducing both the inherent variability in the procedure and, again, the workload for embryologists.
Right, those key benefits.
And he also highlighted the potential for improvements in egg survival and optimizing the timing of the injection, as we touched on.
And Professor Mendizabal-Ruiz, the engineer, he highlighted the role of artificial intelligence, didn't he?
Yes, he did. He explained that with the AI, the system can autonomously select sperm and precisely hold it with that laser, potentially achieving a level of accuracy that, might exceed human capabilities in that specific task.
Interesting.
And he also echoed the expectation that the time taken for the procedure will be significantly reduced as the system continues to evolve.
Right, getting faster. And Dr. Chavez-Badiola, what was his focus?
He focused a bit on how this system compares to previous attempts at automation in this field.
Okay.
He pointed out that the system described in this study is a significant advancement because it automates every single step of the microinjection procedure.
The whole thing, start to finish.
Including the initial steps of sperm handling and selection using AI, which maybe hadn't been fully integrated before.
Got it.
But he also stressed the importance, again, of conducting further studies to thoroughly validate its broader application in fertility treatments.
So overall, it sounds like the experts are, optimistic about the potential, but also realistic. They recognize that there are still steps to be taken.
That's a fair summary, I think.
However, they all seem to agree that this live birth is a really important demonstration, right? That this technology is actually feasible.
Precisely. They view it as a clear demonstration of the feasibility of fully automated and remotely operated intracytoplasmic sperm injection.
Yeah.
Even while acknowledging that further progress in terms of autonomy and efficiency is still required, it shows it can be done.
This whole development really makes you think about the future, doesn't it?
It really does.
This successful birth raises some pretty fascinating questions about how technology will continue to shape these highly skilled medical procedures.
It certainly does. You start wondering, as technology advances at such a rapid pace, how will that balance between automation and human expertise evolve? Especially in sensitive fields like fertility treatment.
Yeah, where does the human fit in? What aspects of this particular development stand out most to you as you consider its implications? Not just for fertility treatments, maybe, but also for other areas of medicine.
Well, anywhere that needs extreme precision and where remote operation could offer significant advantages. Surgery, diagnostics maybe. It opens up possibilities.
What ethical considerations do we need to start thinking about as these systems become more widespread?
That's a whole other conversation, but a crucial one. Access, equity, oversight, responsibility. Lots to unpack there.
Absolutely. It definitely gives us a lot to consider. Well, thank you for taking this deep dive with us today into this truly groundbreaking development in reproductive technology.
My pleasure. It's fascinating stuff.
We hope this has provided everyone listening with a clear understanding of the science, the potential implications, and, well, some of the questions that this milestone raises.
And our aim, as always, was to give you a thorough yet concise overview of this exciting research, highlighting the key findings and the perspectives of the experts involved.
Right.
Without getting too bogged down in the overly technical language.
Yeah, hopefully we struck that balance. We definitely encourage you to explore the study published in Reproductive Biomedicine Online and the related news article if you'd like to delve into even more detail on this fascinating topic.
Definitely worth a read if you're interested.
Thanks for tuning in to the Health Headliner podcast by Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.